Good day, everyone. We are reviewing Wetzel's Limnology textbook. This is edition number 4, and we are focusing on chapter 12, which looks at salinity and ionic composition in inland waters. And today we'll be starting with subsection E, cations ratios. Let's get started. The ratio of monovalent or to divalent cations has been studied in relation to the distribution and dynamics of algae and higher aquatic plants in freshwaters. Early studies, particularly by Provasoli and co-workers, indicated that the MD ratio was significant to the observed growth of some algae. For instance, diatom dominate the algal flora of very hard water lakes, with MD ratios much less than 1.5. However, this relationship was more strongly correlated with the concentration of calcium. There is little conclusive research to indicate that the stoichiometry of MD impacts lake ecology, but MD ratios and divalent cation ratios may be of importance under certain conditions or seasonally due to biogenically induced decalcification in early summer, in calcareous waters, or sequestering of divalent cations by chelation with dissolved organic compounds. Ionic budgets within a drainage basin. The temporal and spatial loading, distribution, and fate of ions vary greatly with the lithology, climatic conditions, drainage, and immunological characteristics and biotic activities of soils, streams, and lakes. Examination of a representative example of an ionic budget from Mirror Lake, New Hampshire, USA, is instructive for evaluating relative proportions of inputs and fates of ions. The ionic budgets of Mirror Lake ecosystem within the Hubbard Brook Valley of New Hampshire was studied for many years. In this drainage basin, nearly all of the precipitation runoff, not lost to evapotranspiration, flowed through soil and was collected in stream flow. At the time of the study, precipitation was acidic, pH 4.1, and strongly influenced by air pollution. Sulfate was found to be the principal ion in precipitation and supplied most of the sulfate that was discharged by the streams. The input of ammonium and nitrate exceeded the discharge of these constituents. The annual deposition of hydrogen ions exceeded that of sulfate and was a major determinant of the pH of the waters of that region. Cationic loadings in stream water greatly exceeded inputs in bulk precipitation, as gains were obtained by weathering, evapotranspiration, biomass, and exchange processes within the soils. Dissolved cations in stream water draining a grading forest remain relatively constant despite extremes in hydrologic output and climatic variations. Particulate loadings and outputs were very small and were dependent on storm peaks. Inputs to the lakes and outputs from the lakes were largely via cation dissolved in the stream water. Regulation of fluxes to the lakes were affected by disturbances within the drainage basins, such as deforestation and loadings from atmospheric sewage, road salt leaching, and other sources of pollutions. For example, clear-cutting forest disturbance resulted in large losses of potassium, in particular to stream water. Of the major cations, potassium was the slowest to recover from clear-cutting disturbances. So before we get into this subchapter 5, let's look at this figure here, or this table I should say. Allocation of elements in % within the Hubbard Brook experimental forest drainage basin. So precipitation inputs, so we can see that sulfur dominates and nitrogen dominates. There's some sodium as well. And then lesser part for calcium, magnesium, and potassium. Net gas or aerosol inputs. So I think this is probably from atmospheric particulate, so nitrogen and sulfur dominates. Weathering release. So this is what releases due to weathering, and that is dominated by calcium, potassium, and definitely not the species that are coming in from precipitation or aerosol input. And then also we have some magnesium here. And then storage or loss, biomass accumulation. So we have biomass accumulates a fair amount of potassium, nitrogen, of course, and then calcium, but it's not really interested in sulfur, even though we have a fair amount of input. Forest floor. So forest floor is not really something that is accepting a lot of elements except for nitrogen. Stream flow, dissolved substances. So what we are seeing in the stream flow kind of reflects a little bit what's going on in the precipitation. We have sulfur, nitrogen, not as much because it's probably uptaken by the soil and the vegetation. Sodium, magnesium, and dissolved substances, and calcium. And then particulate matter is pretty low. Okay. Salinity, osmoregulation, and distribution of biota. Origins and distribution of freshwater biota. The salinity of inland waters is generally very low in comparison to the global oceans, although in some arid regions, the salinity of closed basin lakes can exceed that of seawater by several times. The distribution of biota in freshwaters has been influenced by long evolutionary history of physiological mechanism and adaptation of osmotic regulation to a wide range of salinities. These mechanisms developed against the background of large differences between the salinity in the environment and that of the cytoplasm or body fluids. The general distribution of the freshwater biota and their tentative origins in terrestrial, freshwater, or marine sources are summarized in a detailed review. The distribution of biota in estuaries that interface marine and freshwater environments provides insight into the salinity range occupied by various species. In these transitional waters, as in the spectrum of salinities of inland waters, the salinity range occupied by a species depends on the efficiency of the physiological mechanism by which it is adapted to changes in salinity in the environment. Few freshwater species are truly urea-aline or haline, able to tolerate a range of salinities. In ecosystems threatened by freshwater salinization, the impact of increasing ion concentration on freshwater organisms is complex and is dependent on the magnitude and ratios of individual ions and their temporal variability, the co-occurrence of other abiotic and biotic stressors, and the evolutionary context. Osmotic adaptation of aquatic plants and animals. The function of osmotic regulation is to maintain cellular ion concentration at appropriate operational physiological levels. Most freshwater bacteria and cyanobacteria are relatively homoiosmotic and tolerate only a narrow range of salinity, but can adapt to increasing salinity relatively rapidly by means of genetic change. Extensive adaptive irradiation is seen among these groups. Members of the Protista, algae, fungi, and protozoa with mitochondria, and if they are photosynthetic, with chromoplastids, which are largely single cells, retain considerable evolutionary urisalinity, that is broad salinity tolerance, and are widely distributed with respect to salinity, although some groups, especially among the green algae, are restricted to freshwater. The contractile vacuole is the primary osmoregulatory organism among the Protista. Among the higher plants, only 2% of angiosperms are aquatic, and very few groups extend into saline waters of brackish or marine areas or hypersaline closed basin lakes. So let's see here. Why don't we look at these figures in this table? So table 12-9, average annual Ionic budgets for Mirror Lake in New Hampshire. So it's kind of like a little bit like the same figure we just looked at. When in this case here, we're not seeing actually any sulfur, whereas it's been replaced by protons, by H+, hydrogen. So we are seeing a fair amount of input from chloride, and then sodium. The liter assuming it's the leaf litter, is dominated by calcium and potassium. The dissolve phase inputs is dominated by calcium and sodium and chloride. Particulate, so let's see here. Particulate here is dominated by potassium. Domestic sewage, road salt seepage. It's interesting, we have some question marks here. 4B, and then so it's somewhat estimated, like by difference, I see. And you would guess that we would have a pretty good quantification of that. And then let's see, total inputs. So the total inputs dominated by calcium, sodium, potassium, chloride. Outputs. The most of the output is in the dissolved form, and then in the sedimentation components. So dissolve is dominated by calcium and chloride, sodium, and the likes. As permanent sedimentation, we have a range. We're dealing with a range here. So, and the range is focusing on where we have the most contribution is sodium, potassium, chloride, and then calcium. And then change in lake storage, ultimately the lake is seeing an increase for sodium to a certain extent, and then chloride. All right. Proposed conceptual model for estuarine biodiversity species changes covering the salinity continuum from freshwater to hyper-alkaline or alkaline conditions. So here we have the number of species on the y-axis, and on the x-axis we have the salinity classification. So we have oligoalkaline or alline, we have mesoalline, polyalline, eualline, and hyperalline. So where we are seeing the most diversity is, at the end of the day, is in the marine species here, in the eualline or environment, which I'm assuming to be oceanic. And then the other thing that we're also graphing here, we're graphing marine species, we're graphing estuarine species with those vertical bars, and then diodromous species, which I think means that there are species that can live in either freshwater or saltwater environment. So where we have the least amount of species is in this sort of, mesoalline environment, and at the tail end of the oligoalline, so where the salinity is about 5, and it's probably, I would think, it's 500 milligrams per liter, I believe. So the conclusion at the end of the day, most of the species, the diversity is in the oceanic environment, even compared to the freshwater environment. Nearly all of the higher freshwater animals originated from the sea, although most aquatic insects are of terrestrial origin. Both in terms of evolutionary and contemporary life in freshwaters, osmoregulation is a major problem for which diverse mechanisms have developed to regulate salt and water content. Adaptation to low salinity by some marine animals has been achieved without osmoregulation. Such poikilosmotic animals adjust the osmotic pressure of their body fluid to become more or less iotonic with the salinity of the medium. In contrast, the homoiosmotic animal will tend to retain its initial internal osmotic concentration upon being exposed to modest changes in the salinity of the medium. The general relations between the osmotic pressure among different types of animals expressed as the salinity of the blood versus the salinity of the external water can be visualized in figure 12-13. The range depicted by area A extends over a wide variation in the osmotic pressure of body fluids that is found in brackish water animals, which tend to be more poikilosmotic at high concentration and more iosmotic at lower salinities. The range of the osmotic pressure curve extends from the most homoiosmotic to the most poikilosmotic species. The lower salinity tolerances are more variable, represented by the undefined left-hand edge of area A, but all of the species have failed to colonize freshwater. So figure 12-13, relationship between the osmotic pressure expressed as salinity of body fluids of brackish water organism, A, yellow area, and freshwater animals, C, pink area, and the salinity of external water. Relationship of the curves are detailed in the text. Okay, so y-axis we have osmotic pressure of body fluids in percent per mil. So as you go up the y-axis, the osmotic pressure increases. And then here we have the salinity in percent per mil as well. So as you go on the x-axis, as you go toward the right, away from the origin of the curve, you are in a seawater environment, where at the origin you are in freshwater environment. So here we have the ioosmotic line, which means that it's the line where the osmotic pressure is equal to the salinity, I believe. So first in the freshwater environment, we are dominated by ioosmotic species, and which are osmotic regulators. And then that we have the poikilosmotic, which is an osmotic conformer, meaning by that it conforms the osmotic pressure conforms the salinity of the environment. And then here we have those curves, those yellow curves. So the yellow area is for the brackish water organisms. So this is brackish, and then the pink area is for freshwater species. A few species have succeeded in penetrating freshwaters without a renal osmoregulatory mechanism. These brackish water animals are partially homoiosmotic, area B, which is the blue area we were wondering about earlier on, so which is this area B here, and maintain a very high osmotic pressure in hypertonic body fluids by the active uptake of ions from the freshwater. Excretory organs are not involved, since the urine produced is isotonic with the blood. In most freshwater animals, however, osmotic pressures of the body fluids have decreased to levels equivalent to 5 to 15 per mil salinity to reduce osmotic gradients. These organisms have developed excretory organs that effectively recover ions and produce urine hypotonic to the body fluids. Most freshwater animals, therefore, effect osmoregulation by the active uptake of ions and by a renal mechanism of ion retention. Curves C1 and C2 bound the extremes in osmotic pressures of body fluids in freshwater animals. The isotonic line along the right-hand edge of area C indicates the upper salinity tolerance limit of most freshwater animals, and that they are capable of hypotonic regulation. So let's kind of unravel this a little bit. So curves C1 and C2, they bound the extreme in osmotic pressure of the body fluid in freshwater animals. So it means that that the osmotic pressure of freshwater animals can go up to maybe 22 per mil, and can go as low as maybe 4 per mil. That's one. And then the isotonic line along the right-hand edge of area C. So the isotonic line along the right-hand edge indicate the upper salinity tolerance. So that's the upper salinity tolerance of most freshwater animals. So it's kind of the same that we looked at curves C1 and C2. Although most freshwater animals can live in water of low salinity, the adaptation of body fluids of low osmotic pressure is apparently irreversible, and with few exceptions, freshwater organisms are restricted to salinities of less than 10 grams per liter. Because of the slow diffusion of oxygen in water relative to that in air, the movement of water over permeable membranes or tissue surfaces for respiratory needs is almost universal among aquatic animals. The pumping process places high energetic demands on the animals and additionally exposes cellular surfaces to osmotic gradients. Mechanisms for taking up salts against a concentration gradient vary greatly among freshwater animals. In a few organisms, the incorporation of salts with food may be adequate, but more often, organs have developed for this purpose. Aside from resorption mechanism of the excretory organs, which are advantageous energetically, active uptake mechanism for ions, especially sodium and chloride, are often associated with the respiratory organs, commonly gills, of many invertebrates and vertebrates. The fauna of extremely saline inland lakes is relatively insensitive to the high salinity of these lakes and to large inter-seasonal fluctuation in the chemical composition of the water. Most of the animals of saline lakes are of freshwater origin and include representative of the aquatic insects, phytopods, copepods, and cladocerans, crustacea, and rotifers, all of which belong to predominantly freshwater groups. The blood of these saline inhabiting animals maintain osmotic pressures at level characteristics of those living in freshwaters. The body surfaces of these animals exhibit very low permeability, and they possess effective excretory mechanism for maintaining the body fluids strongly hypotonic to the external medium. Furthermore, the water balance of saline inland waters frequently fluctuates widely. Freshwater animals with resting stages capable of withstanding desiccation have a distinct advantage over marine animals which lack strong development of these characteristics. And that will be it for today, and tomorrow we'll pick it up with the summary of this chapter dealing with salinity and ionic composition of inland waters. I'll see you tomorrow. Thank you. Bye.